All right, so good morning, everybody. I just want to apologize for this morning's minus snafu. Just a few minutes late, just had a couple of technical difficulties, but I just want to say thank you again for joining us for another wonderful webinar series that Bahamas National Trust has been hosting for the past several weeks. Today is going to be even more exciting because we're going to be talking about something that I think everybody is familiar with, and that would be the invertebrates. But before I jump more into that, for those who do not know who I am, my name is Scott Johnson, and I'm Science Officer for the Bahamas National Trust. And I will be your moderator for today's webinar session. Again, we have an amazing bunch of researchers who will be focusing on the largest groups of animals in the country, which are the invertebrates. So just to refresh people's memory who may have forgotten what an invertebrate is, they are normally known as the animals without backbones. And they include things like insects, spiders, snails, crabs, and other animals. So if you ever got stung by wasps, if you ever ran from centipede, if you ever eat crab, all of those are invertebrates. Okay, so today we are in for a treat as our presentations will focus on some of our favorite animals, and that includes the land crabs and insects. So our first presenter will be Dr. Carol Landry, who is the Associate Professor of Plant Ecology at Ohio State University. And her her co-author is Dr. Nancy Elliott, who is the Professor of Biology at Siena College. The title of the presentation is called The Importance of Pollination Ecology in the Maintenance of Coastal Biodiversity in the Bahamas. Why the details matter. So without further ado, Dr. Landry.
Thanks, Scott. I'm pulling up my screen share right now. Come on. Are we good?
Yes, I can see.
Okay, great. Thank you very much for inviting me, Scott, and for moderating today. And a special thanks, shout out to Giselle. To sort of put what I want to talk about in context here, I wanted to share first a quote from On the Origin of Species. Darwin said in this book, near the end of the book, that it is interesting to contemplate an entangled bank, clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and dependent on each other in so complex a manner. Darwin's a lot more eloquent than I tend to be, but I would argue that more than 150 years later, it's still very interesting to contemplate all of these species interactions. And we do so because those species interactions matter a great deal to the survivorship and the reproductive success of all of the organisms on the planet. These days, we refer to any of these kinds of studies in the context. We always put them in the context of fitness. And the details of natural history matter a great deal in terms of the fitness of an organism, which is basically its ability to survive and the relative degree of its reproductive success. This is the biotic component, right? The species interactions form the biotic components of an environment, and they are just as important as whether or not there's enough rain, or whether or not a hurricane approaches, or whether or not it gets too cold, right? All of those things matter a great deal. My colleague Nancy and I have been for over a decade now working together studying plant-pollinator interactions. These are mutualisms, and I'm sure most people are aware that in this mutualistic relationship, plants that rely on animals for successful pollination are benefiting by pollinator availability, and pollinators, which often rely on floral resources like nectar or pollen as a food source, either for themselves and or for their babies, that these relationships are mutualistic. They both gain a benefit. But it's not just about the two of them, that specific pair of interacting partners, right? It gets broader than that, and that's what we're interested in. So this particular study that I'm going to talk about today is part of a larger much larger research program, but it's also the data I'm going to present are parts of different studies that are ongoing. And the main focus is actually on the mating systems of the plants that I'm going to highlight today. And these are the four main questions that I'm going to address. Whether or not flowers are functionally male or female, or if they have both male and female function, which means they're hermaphrodites. And if they are hermaphroditic, are they capable of self-pollination? And will self-pollinated flowers produce both fruits and seeds? If so, then do you make just as many fruits and seeds if you self a flower as a flower that's outcrossed with another individual? And are those seeds equally viable? Now, this is an invertebrate session, and I realize that I am talking mainly about plants, but that's okay, because you'll see that it very quickly becomes much more of a community kind of question or set of questions. So I'm going to share a tale of two plants. These are both very common plants in the Bahamas, throughout the islands. I've been working on San Salvador Island, so that's where this data is from. And Turnera ulmifolia is one of the two species. Erithalis diffusa is the second. I'll present these two, the tale of these two plants back and forth between the two of them in the different categories that I'm going to discuss. So the first question, remember, is what do the flowers look like? What is their gender? And in the case of Turnera, it appears that it is hermaphroditic in the sense that it appears to have fully functional male and female reproductive parts. This particular species, interestingly, across its range, has a variable mating system and a variable breeding system. And so, since this work hadn't been done in the Bahamas yet, this is the first study of the Bahamian population, I'm investigating it here. Erithalis diffusa, also at first glance, the flowers appear to be hermaphroditic. But as I discovered very quickly, when you look a little closer at these very small flowers, some of them don't appear to have pollen in their anthers, which means that maybe they're female. Others have very reduced female structures, and so may or may not be hermaphroditic. They may in fact be functionally male. And so you really have to look very closely when you're doing studies to figure out what even just the floral form is. Now remember, at this point, these are both sets of hypotheses. It's what they look like. And what I did next with pollination experiments is determine what they actually are functionally. Right? So the experiments, the methods that I'm going to describe are methods that were used for both species. There are a few differences that are based strictly on the floral biology of the plants. We don't really have time to go into that. And so this will be just the generalities. So in both cases, there were 4 treatments that were used. Listed here, the auto self treatment is testing whether or not a plant can set seed without visitation by anything. A forced self treatment is when I apply pollen to the female component of the plant, of the flower, using pollen from the same plant. Outcross is using pollen from a different plant, but it's also something that I do myself, hand pollinated. And then an open pollination treatment to see what happens in nature. Right? So the first 3 of these 4 treatments were bagged, the flowers were bagged as buds, so that there wasn't any possibility of an outside influence, another pollinator other than myself. And the second, or the 2 center ones with the asterisks, in those cases, because they were hand pollinated, I emasculated them first. So that means I removed the anthers from the flower before I pollinated them. Regardless of how the flowers were pollinated, once the flower was spent and closed back up, it was bagged, and the fruits and the seeds were allowed to mature, and then collected them and counted. The pictures you've seen here are all for Turnera. That's because it's a much easier plant to photograph. It's much bigger. So I'll start with results from Turnera's study. This is the fruit set data from this study. you can see here that there is a difference between, a significant difference between the auto-self treatment and the other three. I don't think this is necessarily something that might be helpful in terms of nature, although it could be. It was very windy when I was doing these experiments, and I noticed that when the wind blew, the flowers flew within the bags, pressed themselves against the bags, and pollinated themselves. And so without that bag around it, I'm not sure that there would have been fruit set automatically. In other words, without any kind of visitor, including myself. The other three treatments, though, there's no significant difference between. And that's true whether or not I consider the autosols. The seed set for that same study now, when I look at this, you can see that the forced self and the outcross treatments, let's just look at the blue bars first. Those are viable seeds on average per fruit. And the two hand-pollinated treatments differed significantly from the open pollinated treatment. Additionally, I looked at aborted seeds or aborted ovaries, sometimes they're classified together, and saw that there was significantly more of those aborted seeds in the open than in the others, right, than the other three treatments. So what can we take away from this? The flowers do appear to be hermaphroditic. They selfed, or I could self them, and they're self-compatible. Auto-selfing is possible, but there would need to be some sort of mechanical method because I don't know if the pollen would be transferred properly otherwise. And because there is such a great reduction in viable seeds in the open pollinated treatment relative to the others, it could be limited by the number of pollinators that visit, but there are other possibilities as well, right, which I'll get to in a minute. In terms of Erithalis' pollination experiment, all I have is fruit set data for that. You can see that there is a significant difference between either of the self-treatments versus the other two. And that means they are not self-compatible. And because there is just as much fruit set in the open pollinated treatment as there was in my hand pollinated outcross treatment, it does not appear that pollination is limited by the number of pollinator visits. Whether or not there are multiple floral types, that is an ongoing study. So in this particular study, I used only flowers that appeared to be hermaphrodites, where they had a full-sized female structure as well as pollen in the anthers. So I haven't completed those other tests. Now, how do these plant-pollinator interactions influence the community-wide biodiversity in coastal communities? Let's look first at their visitors, floral visitors, which are potential pollinators. This is a lovely image of a Megachile female who's showing off her pollen load here. She's alive, by the way. I think that there are 9 different insect families represented here. The 2 bee species at the top, Megachile and Xylocopa cubicola, are both well known as very common generalist bees. I have very limited observational data on how efficient these different species are because this is ongoing, remember. Xylocopa visits seem to result in a large number of pollen deposited on the female component of the flower, making it a very good pollinator. But in contrast, species like Agralis, which is the Gulf fritillary Lepidopteran, that left very little pollen grains on the female parts after they leave. So whether or not they're actually pollinating flowers when they visit is, you know, a question. In addition, I noted during this particular study a number of other relationships with this plant. The shield bug is a sap-sucking insect, so an herbivore. It's in the upper left corner. The little fruit next to it has been invaded by a seed predator of some form. I found 5 different morphospecies representing at least 3 different insect families inside the fruits. And that means both in terms of larvae, pupal cases, and mature insects inside the open pollinated fruits. The flower that has been cut up looks to me like it may possibly be a megachilid cut, using this to build nest cells. And down below, this is an interesting plant in many ways. It appears to be ant dispersed in terms of the seeds. And on the lower left is a picture of ants inside the fruits. They gather them, they eat this tiny little piece of protein that's attached to the seed and carry the seed away. And an insect gall in a plant here with exit holes in the lower right. So this is just like over the course of a month and a half, I saw these other kinds of interactions which are supporting other organisms, not just pollinators. In terms of Erithalis, there's a much larger number of organisms that visit the flowers. There are 10 different insect families represented here in this list of, I think, 12 species. And banana quits, which were surprising to me because the flowers are so tiny. This photograph is of the Eutoida agizia, which is the Mexican fritillary visiting flowers. Now, beyond these plant pollinator communities, which is all I've really spent much time talking about, and of course the other relationships with Turnera, what's the broader impact on biodiversity? Well, you take one step back, or depending on how you want to think of it, in the trophic chain, and you start to see that entangled bank showing up again. We've got, rock, the iguanas that are herbivores, generalist herbivores. They graze on plants, and that grazing activity tends to stimulate the plant to create branching and new growth. My tiny little praying mantis friend is a major insect predator, eating other insects. And then this lovely photo that Lee Newton graciously allowed me to use of a green heron eating a cicada. Green herons will eat almost any invertebrate they can put their beaks around. And so they are all interconnected. And without that base plant pollinator community, you lose a lot of other organisms or reduce their likelihood of survivorship by reducing the biodiversity at that more basic level. On that note, I'd like to thank the BEST Commission for permission to do the work, the GRI at the University of the Bahamas, where I use as my base, and of course the people of San Salvador Island, who have been gracious and helpful in many ways. Funding sources include the Fulbright Core Scholar Program and my institution, and then also Lee Newton. Thanks very much for the use of her photograph. And I hope I'm within my 15 minutes.
All right. Thank you so much, Dr. Landry. That was a wonderful presentation. I do have some personal questions that I would like to ask, but I will wait until afterwards to do so.
Sounds good.
Before we begin with the second presentation, I just want to remind everybody that the session is being recorded and is being streamed live on Facebook. So after today's session, the recording will be available to all by visiting the Bahamas National Trust Facebook page and YouTube channel. So if you would like to keep up or get in touch with the Bahamas Natural History Conference, you could do so by visiting our website, bnhc242.com, and facebook.com/BNHC to follow social media updates. Any questions and suggestions that you may have can be emailed to bnhc@bnt.bs. So our next presenter is Ms. Tammy LaPalooza of State University of New York with associations with College of, I'm totally gonna mess this up.
Oneonta.
That's it. Thank you.
Oneonta, yep.
Thank you so much. Okay. So with that being said, Tommy, you want the floor?
Yep, I'm going to go ahead and share my screen now.
Okay.
Oh, let's see. Okay, so we should be all set then?
Yes.
You can see it? Okay, great. So good morning and thank you to the Bahamas National Trust for hosting this virtual offering of the Bahamas Natural History Conference. As I begin my presentation, I really want to express the gratitude to the land crab harvesters I've met over the years. This project really grew from my fascination with the land crab harvesting I initially learned about more than 25 years ago, both as a college student studying the botany of the Bahamas and then as an intern at Forfar Field Station on Andros Island. I did receive partial funding for this project through the SUNY College at Oneonta, and I also want to spend a special thank you and shout out to Carl and Cattell Roll at Roll's Place Cabanas and Diane Adderley at Adderley's Grocery in Fresh Creek for providing support during the 2019 research expedition. Today, I'm going to talk about the research we've been conducting over the past decade, with a focus on the more recent efforts to conduct a rapid assessment of the Crab Replenishment Reserve. Our goal is to collect data that will affect management considerations of the Crab Replenishment Reserve, a national park in central Andros, with significant cultural importance for the Bahamas and the people of Andros Island, who have provided for their families for generations through harvesting land crabs. The main premise of this ongoing project has always been to find a way to support continued harvest at a level that will maintain quality of life while leaving a sustainable population of land crabs to harvest into the future. So the title of my talk today is Rapid Assessment of Crab Replenishment Reserve and Perspectives for Management of a Culturally Significant National Park. So first, what is a protected area? So the International Union for Conservation of Nature defines a protected area as a clearly defined geographical space, recognized, dedicated, and managed through legal or other effective means to achieve the long-term conservation of nature with associated ecosystem services and cultural values. And so protected areas go by many names, some of which include wilderness areas, community conserved areas, nature reserves, and national parks. The efforts to establish protected areas play an important role conserving nature and the services it provides us, namely food, clean water supply, medicines, and protection from the impacts of natural disasters. The importance of these spaces as we move forward with the effects of climate change and the current pandemic cannot be underestimated. Protected areas in the Bahamas are managed by the Bahamas National Trust, an organization established through an act of parliament in 1959. And the Trust's mission is to effectively manage the national parks within the archipelago. The Protected Area Network has expanded to 32 national parks and counting through a vision to safeguard biodiversity while also providing areas for recreational enjoyment. The Bahamas National Trust relies on volunteers and donations to meet the ever-expanding needs of being the caretakers, protectors, and voice of the Bahamas' most precious natural resources. This not only includes management of these unique spaces, but also engaging in scientific research to monitor impact, and more importantly, engaging learners in the ultimate outdoor classroom experience. Effective management of the national parks is enhanced through the many science-based initiatives and partnerships with organizations working toward the shared mission of conserving and protecting Bahamian natural resources. And so the Crab Replenishment Reserve was established on North Andros Island in 2002 with the goal to forever protect not only land crab habitat, but also a culturally significant space in central Andros, where land crab harvesting has occurred for generations. Contained within the park is habitat utilized by every stage of the land crab life cycle, from inland evergreen forests where adult crabs create burrows to access the water table, to coastal areas where female crabs deposit their eggs during the late spring and summer spawning migrations. Spawning events are more numerous after a heavy rain, creating pockets of fresh water where the crabs can moisten their gills during the migration to the sea to lay their eggs. It is during the spawn that the bounty of the land crab harvest can be seen. Innumerable crocus sacks, which are bags used to hold the hand-caught crabs, are loaded into boats to be shipped not only to the main ports of Nassau and Freeport, but throughout the Family Islands so that people can enjoy the flavorful delicacy of the land crab. Occasionally, folks will transport their land crabs in boxes or coolers via airplane, and we are entertained with videos throughout social media that document the escaped land crabs taking a spin on the luggage conveyor belt. The Crab Replenishment Reserve is not the only location throughout Andros that is frequented during the spawning migration. Harvesting in this historically important site has never been restricted or prohibited, as generations of Androsians have provided for their families during the seasonal abundance. The cultural nuances of the land crab fishery and the availability of the land crab as a food source are so important that an annual festival is established to celebrate the land crab heritage. So our initial studies began when we sought to gain an understanding of the genetic diversity of the land crab populations. We then really wanted to analyze the harvest trends to start to determine the size and sex ratio of the land crabs that are being harvested annually. We did find a harvest ratio of 2:1 males to females, which could impact long-term sustainability if the female land crabs are harvested prior to releasing their eggs. We still do not have data to understand the population density of land crabs on Andros, or to be able to calculate the standing stock of land crabs. This led to conducting rapid assessments of the land crab populations on North Andros Island. It is important to note that there are actually 2 species of land crabs that are harvested for food. And so the black land crab was present all throughout all of our research expeditions. We did not actually focus on that for our current project. And so in 17 was really our first expedition. And so we brought down a small group of undergraduate researchers from my college, and we also had a few BNT interns that were working with us. And we did present the results of our 2017 pilot study at the BNHC, which is actually available on the BNT's YouTube channel. So if you'd like more details on that part of the project. What we did find was that the land crab population density really depended on the habitat, which was actually what we expected based on the life history of the land crab. However, our calculated population density was low compared to the 1983 reference study. And that 1983 reference study was one of the only other larger scale population surveys on Andros Island. And so clearly more data was necessary. And the reality was that as we began to sample the habitat and the population density in the Crab Replenishment Reserve, it became clear that a separate expedition focusing specifically on surveying the population of land crabs in the national park would be necessary. And so in 2019, our expedition really did just focus on surveying the national parks, and we had 2 goals. The first was to survey the type and abundance of habitat that was useful to the land crab at various life stages, and then also to conduct a rapid assessment of the density of land crabs in each habitat type. And so knowing that the Crab Replenishment Reserve is 4,000 acres, our plan was to cover as much area as we could, knowing that some of this habitat would be inaccessible. And so we established a grid to really just evenly distribute the transects along the length of the park. And so we set the first and the last transect equidistant from the north and south borders, and then we distributed the 12 transects within the park 500 meters apart. And so we did survey as much as reasonably possible within each transect. The forest is rather dense, and so we ended up using compass bearings to keep us due east or west as we counted active land crab holes within a 2-meter belt along each transect. And so the active land crab holes were visually determined by either presence of a live crab or scat that was adjacent to the hole. Fenella, who is one of our students, one of my researchers from the 2017 study, returned as a volunteer to assist in 2019. She definitely mastered the art of shining a light into crab holes to confirm the presence or absence of a land crab. And so the habitat types that we looked at, the first 2 were looking at the Pineyard and the Evergreen forest. And so the habitat was interesting to say the least and contained the typically expected plant species for each habitat type. Much of the Pineyard had a dense understory, including lots of palms and an abundance of poisonwood. The Evergreen forest consisted of similarly dense understory with a few open spaces, which made access impossible at times. Interestingly, we did encounter a section that looked thinned out, possibly someone making a trail, and then there was another section that there was definitely a trail. However, this was actually a wild boar trail that was creating space to allow us traverse low to the ground. We did actually know this was a wild boar trail because we stumbled upon an intersection of trails with fresh boar scat in the middle, and that pretty much ended our route on that particular transect for obvious reasons. And so when we were surveying the scrub habitat is really this shrubby dry habitat, and the density within the shrub varied depending on some of the areas within the park. And so if you've not had the opportunity to explore this unique space, know that this is a very special habitat, and it's predominantly poisonwood, jowwood, thorny shrubs and vines, and most of the vegetation is no more than 2 meters high. The terrain consists of an assortment of holes with sharp, jagged edges, some deep enough for a leg, others fit for a person. This also made our progress slow because these holes are a favorite for the land crabs, and it seemed that in each hole we encountered one. And so this is just a video showing what some of that terrain looks like. And again, there's large swaths of this scrub habitat throughout the national park. And so we estimated the amount of each of these main habitats so that we could then extrapolate the abundance of land crabs in the preserve. So the broadleaf forest and pineland is really concentrated in the north and east edges. So it would be these sections right here. A lot of this is just evergreen forest, and there's some pine forest in here. And this is all mostly broadleaf forest as well. Approximately half of the habitat is scrub, and the lower section of the scrub is less dense than the northern section. And there's also evidence of ephemeral flooding in the lower section, so it would be in the central part right in here and along the Queens Highway. And we assume that there was some ephemeral flooding because there were many shallow pools of fresh water. There are also red mangroves found quite a distance inland as either individual plants or small patches, which are likely transported there as propagules during storm events. Logistical challenges did prevent us from accessing the lake habitat in the northwest section. And so this, we believe, is a lake area. However, we were able to access the eastern edge of the blue hole in the middle southern area of the park boundary. So there's a large, sizable blue hole right here along the edge. And so along this northeast edge, there's actually quite a large swath of red mangroves stretching out pretty much as far as we could see. And there's also this very marshy habitat that surrounds the blue hole. So again, right in this area here. And additionally, there's also a pineland ridge on the back side of the blue hole. And so really think it would be important to follow up for an expedition to really assess that lake area and that blue hole to really document how the land crabs are using this space. And part of that is because I have been told by land crab harvesters on Andros that they have seen land crabs spawn in the freshwater lake spaces on the island. And that's interesting because generally speaking, the land crabs will only spawn in salt water. And that's actually the only stage of the land crab life cycle that requires salt water. The rest of it is all freshwater. So again, just a really interesting perspective from one of the land crab harvesters that's worth further exploration. And so our data really just confirms what we expect to find based on our understanding of the land crab life cycle and how the land crabs utilize the different habitats. Each individual female land crab will release hundreds of thousands of eggs. However, only a small proportion of those reach adulthood. And each stage, thus age class of the land crabs, utilizes the different habitat types. And we would expect that as the age class increases, the abundance of crabs would decrease. And as expected, the highest density of land crabs was found in that scrub area, which is incredibly popular and the most abundant space where you'll find the juvenile crabs. And so it's also interesting that even though there is approximately the same proportion of Pineyard and Evergreen forest in the reserve, we did find a higher density of land crabs in the Pineyard. And this is likely due to the numerous holes in the limestone substrate that provides ready access to the water table. In contrast, crabs that inhabit the evergreen forest need to dig their own burrows to access the water table. So it's definitely easier to find adult habitat in the Pineyard for the land crabs. And so then we estimated the standing stock of land crabs in the national park. And we found that to be about 5 million individuals. Only about 1 million of those are expected to be harvestable size. And so these would be the adult crabs that would be found within the broadleaf forest and in the Pineyard. And again, this abundant juvenile crab habitat really just provides important nursery ground for the growing crabs as they move into the adult crab habitat of the Pineyards and Evergreen forest. What we don't know is how many crabs are harvested from the Crab Replenishment Reserve each year. And so determining the level of sustainable harvest is challenging, but an important component of managing not only the national park, but also the land crab fishery across the Bahamas, which is valued at $20 million US. And so when we're thinking about, how to manage this space, for national park tourism and other means, there are really some unique features that I think could be incorporated into the structure of this tourism. That blue hole is certainly interesting. I'm not sure this would not be for the average national park tourist. This is definitely more adventure travel. And so it's quite a distance back there, but again, it's just a really, really unique feature to this wonderful protected space. And we also happened upon more than one wild boar trail. And so we also found what would be likely an ephemeral pond that there's definitely wild boar hair found all around it. And so likely the wild boars were using that as a watering hole. We were also told that the Replenishment Reserve contains a few areas where persons will hunt either for wild boars or ducks or other hunting prey. And so other activities that would attract the ecotourist would of course be birding, birding for looking at the birds and studying their ecology and some of these magnificent creatures. I actually encountered a great lizard cuckoo, and I wasn't sure if it was checking me out as a meal or just wondering what I was doing in its part of the forest. And there's also a really wonderful mangrove estuary expanse on the eastern edge of the park. And this leads to a really, really beautiful shore space. And piping plovers are known to nest there as well. And so of course, any part of creating trails could really highlight some of the land crab life cycle and the different plants that are found throughout the Bahamas. There really is some magnificent forest contained within this national park. But regardless, the Crab Replenishment Reserve is a well-known location for harvesting land crabs. And through partnering with the local tourism board and other community partners, the local land use structures could be incorporated into successful management of this protected area. One of the main highlights of the land crab fishery and certainly tourism on Andros Island is certainly Crab Fest. And so you can't talk about the Crab Replenishment Reserve without talking about Crab Fest. And so the All-Andros Crab Fest was established and organized by the local government and tourism board in Central Andros as a small gathering at Queens Park in Fresh Creek, Central Andros, to celebrate the legacy of the land crab harvesting. The 24 years since these small beginnings, the All-Andros Crab Fest has grown into a 3-day event, which brings together both domestic and international tourists to Fresh Creek, Central Andros for a weekend of cultural activities. There's something for everyone at the festival, which begins with a church service on Thursday evening throughout the weekend to highlight entertainers. There's a market with goods for sale and also food vendors serving a variety of land crab dishes and other local cuisine throughout the weekend. The festival merges into the local community, where family reunions and community gatherings fill the beaches with festivities. Hotel and restaurant operators throughout the island are at capacity. In recent years, day-away excursions on the fast ferry have provided the opportunity for people traveling from Nassau to bring not only themselves, but also a vehicle to facilitate travel during Crab Fest. To understand the scope of what this event brings to the economy of Central Andros, realize that the resident human population of North and Central Andros combined is fewer than 5,000 persons, and it's estimated that during some years more than 20,000 persons have traveled to Fresh Creek to experience the All-Andros Crab Fest weekend. To say that Crab Fest is the place to be in June is an understatement, and I think certainly the impacts of the current pandemic causing the cancellation of not only the Crab Fest and other national heritage festivals will be felt for quite a while in some of these small communities. We're definitely looking forward to Crab Fest 2021. And so, when we're thinking about the cultural significance of the land crab festival, the land crab fishery, really preserving this legacy of the land crab harvesters beyond just the Heritage Festival, I think could really be accomplished by documenting all of the local ecological knowledge that the land crab harvesters have acquired over the years. And, you know, this is definitely, I think, where the richness and treasures of a lot of these heritage festivals and certainly heritage fisheries begin. And so this local ecological knowledge is incredibly important because the land crab harvesters have a unique understanding of their fishery resource. And so, you know, I really think that as we move forward, this, you know, having this understanding, because as this harvest pressure increases and climate change continues, we're seeing global ecosystems being altered. And so the land crab harvesters' knowledge of their traditional fishery methods and this local ecological knowledge of the land crab itself is really going to be crucial moving forward. And so some of the other perspectives for managing really, you know, including how the land crab fits within the food web of other organisms and not just humans. And so, you know, the land crab really fills a few different niches within the food chain. Certainly, you know, food for, you know, the juvenile land crabs and the eggs provide a bounty of food for other organisms. The adult land crabs generally think of themselves occasionally at the top of the food chain, especially if you encounter a large male. He definitely would want you to think that he is at the top until you throw him into a pot of crab and dough. And additionally, you know, the growing network of the national parks really function in unison to conserve a multitude of species. The West Side National Park is a prime example. And so this protects really a vast swath of Andros Island. And so this is really nursery habitat for hundreds of aquatic and terrestrial species. And it's also an important adult habitat for many organisms as well, including the endangered rock iguana. And so these long-term studies of the national parks will be even more important moving forward to document these changes and adjust management strategies as the impacts of climate change are realized. Now that it's almost lunchtime and folks are starting to get hungry, we're really thinking about food for the past, present, and moving forward into the future. And so managing these fishery resources is certainly not without its challenges, especially when a resource has been historically abundant as a readily available food source. There is no special equipment necessary to harvest land crabs, other than a sack or other container to keep the crabs if you don't want to hold them as you're walking home. Hand catching land crabs is a simple process, and people have been utilizing land crabs as a food source for hundreds of years. Currently, the Bahamas does import 90% of its food. And so this present challenge is not only for the land crab fishery, but all locally acquired food sources, protected or not. And there are initiatives in place to really work on reducing food imports of both plants and animals. However, the current pandemic has put these on hold. Food security has certainly increased. And when combined with the disruption in the food distribution network, the challenge is real when persons are trying to feed their family during a pandemic. And so moving forward, sustainable harvest of the land crabs into the future will depend on determining the maximum sustained yield. That is, determining the highest possible annual catch that can be sustained over time. And this is the cornerstone of managing fishery resources. But for now, the appropriate management of the crab replenishment reserve, along with improved distribution of this fishery resource, could support the continued harvest at a level that will maintain quality of life while leaving a sustainable population of land crabs to harvest into the future. And so I really want to thank all of the land crab harvesters for sharing their rich knowledge of this incredibly important artisanal fishery resource. And certainly all of the field researchers over the years that have helped me in this project. It's not easy traversing through many of the habitats. And so a few of these have moved on. I've even, corralled my own offspring into helping me with a lot of these projects. But certainly, Shannon Yates and Fenella Campanino, they're both moving on to graduate work, looking at different fishery challenges and other species that are important within ecosystems. And so again, just my deepest gratitude to all of the land crab harvesters that I've met over the years, and certainly thank you to the Bahamas National Trust and the Best Commission for permitting me to work on this very important local resource and within the national park.
All right, thank you so much, Tammy, for that wonderful presentation. It is also very concerning. saying how the land crab is such a significant part of the Bahamian culture and the Bahamian economy. So before we move into the next, presentation, I would like to inform you all about the recently published Natural History of the Bahamas Field Guide. And so the Natural History of the Bahamas Field Guide is small enough to carry on trips, and it has an ultra comprehensive guide of terrestrial and coastal flora and fauna, including species and common names, locations, and detailed descriptions. The book was written by experts in Bahamian ecology with contributions by more than 30 other naturalists and researchers, and covers organisms studied in the Bahamas and the Turks and Caicos Islands with more than 700 color photos. It is available for sale in New Providence at Logos Bookstore and Media Enterprises, as well as all national park offices that the BNT manages throughout the Bahamas. The book can also be purchased online at Cornell University Press and online book retailers such as Amazon and Kindle, instantly and by mail. All royalties from book sales benefit the Bahamas National Trust, so it is a great way to support the BNHC. All right, so with that being said, our next speaker is Dr. Deborah Matthews of Florida Museum of Natural History, and her presentation is entitled Birds of a Smaller Feather, a look at plume moths, Lepidoptera, and I'm not going to try this next word, in the Bahamas. So without further ado, Dr. Deborah Matthews.
Hello, everyone. First, I'd like to thank Giselle and the Bahamas National Trust for the opportunity to share a small part of our work. Our surveys cover all Lepidoptera, and thus far we've recorded more than 1,000 species over the last 9 years. We have about 71% of these identified to species level, so there's still a lot to do to document the entire fauna. Within the Microlepidoptera, there are many new species to name and describe, and this often requires help from taxonomic specialists of the various families. I happen to be a plume moth specialist, so it was my pleasure last year to publish an account of the 23 species we've encountered, and I will share some examples of these today. The family Pterophoridae includes about 1,413 species worldwide. These tiny mosquito-sized microlepidopterans have long spindly legs and wings divided into feather-like lobes or plumes, hence the common name plume moth or featherwing. They've also been referred to as T-birds, T-moths, or Jesus Christ moths, based on their characteristic resting posture, with the wings outstretched perpendicular to the body. Our field surveys have been a team effort with different people having worked various islands, and often a group of 4 of us going out for a week or more at a time. We each have different taxonomic interests, but all work together to collect and photograph representatives of each species encountered. We go out scouting localities and larval host plants during the day, but also spend a lot of time each night in front of a collecting sheet. For plume moths, it's been the most productive, however, to wander around with a net and headlamp at night to spot these tiny moths perching on vegetation, nectaring, or hovering around the larval host plants. Setting up a sheet like this does attract attention from time to time and often requires an explanation. There are many reasons for studying Lepidoptera, but the most important is that we need to know what species are present now as a baseline for assessing the impact of environmental changes. We also remind people that butterflies and moths can be important pollinators, food for other animals, plant pests, bioindicators, and educational tools. The Lucayan archipelago is comprised of more than 700 islands, including the Commonwealth of the Bahamas and the Turks and Caicos. We've surveyed 13 of the major islands, but still have not collected moths from Acklands or the Turks and Caicos. The green dots show the number of pterophorid species from each island. These numbers are not necessarily indicative of the total diversity, since some islands have been visited multiple times. Grand Bahama was especially productive, with 12 species found in one 5-day trip, including one of the new endemic species. The more remote islands, such as Great Inagua, have yielded fewer species, but also include endemics. More sampling would be nice, but the logistics of working some of these out islands isn't easy. We try to sample a variety of habitats with both day and night collecting. These are some examples of typical habitats. On the western islands, these include Caribbean pinelands, but all the islands have coppice, beach, mangroves, and disturbed habitat. There are different types of coppice, but these are basically secondary dry forests with various shrubby trees such as mahogany, gumbo limbo, acacias, poisonwood, and sea grapes. It's often difficult to distinguish habitat types because they intergrade together, and disturbed sites such as dumps and old homesteads seem to have the greatest diversity of herbaceous plants and are thus often the best collecting. My favorite habitats are coastal rockland and beaches, where you see dense patches of a ferny-leaved composite, Ambrosia hispida. It's a ragweed, also known as bay geranium. It's found throughout the archipelago and is the larval host plant for one of the most abundant plume moth species, Dijongia californicus. Once you have the search image down, these larvae are quite easy to spot because they're bright red against the green leaves and shoots where they feed. The last instar larvae are a little harder to spot because they turn green before pupating. Populations can be quite heavy, with several larvae infesting the same shoots, and with such dense populations, it's not surprising to see pupae that have been parasitized by wasps. Here's some close-ups of these creatures. The adults are quite small, with wingspans not much more than a centimeter. They cling to the plants when it's windy, and I found the best place to spot the adults at night is in the taller plants protected behind sea oat dunes. The larvae are quite remarkable in that they have glandular setae which produce a sticky exudate. In addition to leaf shoots, the larvae also feed on developing flowers and pollen. The pupae are also very striking since, like a lot of pterophorids, they have both primary and secondary setae. Here's a pinned adult. We have this species from 6 islands, but I suspect now that we know what to look for, it will be found throughout the Bahamas. The bold wing patterns make this a good flagship species for the islands. In addition to the Bahamas, the species ranges across the southern United States all the way to California. There are, however, some differences between Bahamas and mainland populations. Male and female genitalia are identical, as are sedal patterns, but there are 3 main differences. First, the ground color of Bahamas specimens is dark gray as opposed to brown. Second, Bahamas specimens are noticeably smaller. And third, the red spot on the second and third abdominal segment of pupae is not as well developed in the Bahamas. CO1 barcode analysis places the Bahamas specimens within the same species as U.S. specimens. However, a closer look at haplotype networks across the species range may further delineate population segregates. Before moving on to some other examples, I want to point out how challenging it is to work with such tiny moths. Most of our time goes into preparing and identifying specimens after returning to Florida. While it's possible to pin specimens in the field, we've found it's best to layer and pack them in dry tissue, and then rehydrate and pin them under a microscope using small spreading boards. Each specimen is labeled with full data and a unique identifier number before being sorted into morphospecies. For some specimens, it's necessary to dissect the abdomens and prepare genitalia slides in order to identify the species. Most importantly, specimens are stored in airtight insect drawers in climate-controlled conditions to prevent mold and pest damage. Next, I want to show you some more examples of Bahamas species. This is the lantana plume moth, Lantanophega pusillodactyla. Larvae feed and pupate inside the flower clusters, and the adults are easy to find hovering around the plants at dusk. It's common throughout the islands and is basically a pan-tropical species. There are 7 species of lantana in the Bahamas, including 1 endemic. Since Lantana camara is a pasture weed toxic to cattle, this moth was studied early on as a biological control agent and introduced from Mexico to Hawaii in 1902. This is Halenzia paleaceus. It's common throughout the eastern U.S. and larvae feed on the composite genus Vernonia. So far, we've only encountered these once. It was on North Abaco where we set up lights in a recently burned section of pinelands. Hardly anything came to the sheet that night, but I was fortunate to find several adults nectaring and perching on Vernonia blagedii. This is Adana simplicius. The larvae feed within the flower heads of Neurolena lobata. This shrub is common in pinelands, coppice, and especially disturbed sites. The adults fly around the flowers at night. We've seen the plants in flower from March through October, but this is really dependent on rainfall. These flowers are also especially attractive to butterflies as a nectar source. The same plant is also a larval host of Adana ambrosiae. In this case, the larvae feed on the leaves. You can easily recognize past damage because there's a larva-sized resting groove chewed out along the leaf midvein, which is surrounded by feeding damage. It's unusual to find more than 1 larva per leaf, but in this case you can see 3 larvae along the midvein. The larvae have some pretty impressive barbed lateral setae that fan out from lobes called verrucae. And here you can see a larva actively feeding while still hanging out in its groove. There's many composite hosts recorded for Adana ambrosiae in the U.S., but in the Bahamas, besides Neurolena, the larvae also feed on Melanthera aspera and a yellow-flowered Bahamas endemic called Wedalia bahamensis. In this slide, you can see other insect damage on the plants, where the leaf cuticle is completely consumed instead of skeletonized. Here's yet another species of Adana, Adana perplexus, which feeds on Melanthera aspera. Larvae skeletonize the leaves but do not make a resting groove and have much shorter lateral setae. The host plant in this case is growing along a beach boardwalk and is a common plant of disturbed areas. Post-Platyptilia flinti was a complete surprise on Grand Bahama. I collected a series of adults in a roadside ditch that at the time I thought were either Stenoptilodes brevipennis or Taprobanes. As it turns out, the series included all 3 of these very similar looking species. They were all flying around a large patch of Scutularia havanensis at night. Only after dissecting the genitalia and taking a closer look at the adults, I realized that flinti can be distinguished from Stenoptilodes by having cream-colored metathoracic patches. Flinti was previously known only from Argentina, Brazil, and Paraguay, so it's kind of a mystery how it ended up in the Bahamas. The larval host is unknown, but Scutularia is a likely candidate given that other post-Platyptilia feed within the plant order Lamiales. Sometimes stopping for a cold drink after a hot morning of collecting can have unexpected benefits. In this case, a new record for the islands. We found this patch of Borhavia diffusa in the flower beds of Smitty's convenience store on Great Exuma. It was loaded with adults, larvae, and pupae of Megalorrhipida leucodactylus. These larvae bore into the sticky fruits of this weed and consume the tender ovule inside. Here's a live adult perching on the host plant. This species is pantropical and also feeds on several other Nyctaginaceae around the world. It's basically a common lawn and disturbed habitat species. This is Lioptilotes albistriolatus. In the U.S., larvae feed within composite heads of several genera. In the Bahamas, thus far it's only associated with Baccharis flowers. There are 4 species of Baccharis in the Bahamas. This one I believe is Dioica from All-Andros. The genus Exelastus is associated with the family Fabaceae. Exelastus pumilio feeds on Desmodium, also known as beggarweeds. The larvae feed on flowers and leaf shoots. You may recognize these from the seed pods that stick to your socks. There are some native species that occur in the pinelands, as well as Desmodium incanum, which is another common lawn weed. In the image above, you can see the little green pheromone gland of the female is everted at the tip of the abdomen. Most female plume moths have these dorsal glands to call in the males. This one is Exelastus montes Christi, another legume feeder, in this case on Rhynchosia minima, which also grows in lawns and climbs fences. In 2008, Bernard Landry and I described Exelastus dowi from the Florida Keys, Belize, and a single 1980s specimen from Grand Bahama. I'm happy to say we found more specimens on Grand Bahama, as well as South Abaco. The host is still unknown, but probably also a legume. It can be distinguished from Montes Christi by having dark spatulate scales along the entire length of the hindwing anal margin. I've shown you some examples of mostly common species. Now I'd like to introduce you to our 4 new Bahamas species. These are known from very few specimens, and although I would have preferred to have longer series, I thought it best to go ahead and get these described after having reviewed the known neotropical fauna. Halinzia leucayana is from Crooked and Mayaguana Islands, Halinzia bahamensis from Grand Bahama, and Edematophoros androsensis from All-Andros. Michelaphorus silensis is known from Crooked Island and Great Anagua. Since sea salt production is the main industry on Anagua, the specific epithet is derived from the Latin sal for salt and the root ensis meaning of or from. Nothing is known about the life histories of these 4 species, although for Halinzia and Edematophoros, the larval host plants are most likely composites. Here's the list of species found so far. With the exception of only 5, all of the Bahamas plumes also occur in Florida. As our knowledge of the Cuban fauna becomes more complete, most of these species will probably be found there as well. In terms of habitat associations, nearly all of these species can be found in disturbed habitats. This is because pterophorids feed mostly on opportunistic weedy species. A couple species seem to be more associated with pinelands, such as Exelastus dowi and Halinzia paleaceus. No species is restricted to coppice, and only Dijongia is strongly associated with beach and coastal rocklands. No plume moths are strictly associated with mangrove habitats either. Looking at host plant relationships, as expected, more than half the species are associated with composites and 4 with the order Lamiales. This corresponds to the worldwide pattern of about 40% Asteraceae and 20% Lamiales. Although I know the hosts for several species based on what they feed on in Florida, there's still work to be done in finding the actual hosts used in the Bahamas for a few of these, and of course for the 4 new species. I want to thank our field team for all their contributions and note that along the way we've made lots of friends that have facilitated our work on each island. These people have sometimes accompanied us in the field to learn more about Lepidoptera in their own backyards. I'll end by noting that we occasionally run into many-plumed moths. This is the family Alucididae. Jean-Francois Landry of the Canadian National Collection has barcoded our specimens, and we expect these will turn out to be another new species for the islands. Thank you for your attention, and again, thanks to the Bahamas National Trust for the opportunity to present this part of our work.
All right. Thank you so much, Dr. Matthews, for that wonderful presentation. It's amazing to know how many different species of plume moths there are in the Bahamas. And I find that very, very intriguing. We are going to move into our final presentation. And our final presentation is going to be, one second. Is going to be by Mr. William Bigelow of Memorial University of Newfoundland, and who's working in partnership with the Cape Eleuthera Institute. His co-author is Dr. Ian McGaw of Memorial University of Newfoundland and Department of Ocean Sciences. And his presentation is the physiological and behavioral responses to dehydration in the black land crab, Gecoscinus rubricola. So without further ado, Dr. Bigelow.
Hi, good afternoon, everybody. How are we doing? Yeah, so can you see the screen okay?
Second. Yes.
Okay, perfect. And you can hear me all right? Perfect. All right, well, good afternoon. Thank you for the introduction. My name is Bill Bigelow, and as mentioned, I am a graduate student at Memorial University based in St. John's, Newfoundland. And my project, my master's thesis, is taking a look at different ecological and physiological aspects of the black land crab here in Eleuthera. And for today's conversation, I'd really like to elaborate on some of the deficits in our knowledge of this species and different ways that we can expand that knowledge, both here in Eleuthera as well as throughout the Bahamas. As Dr. Tammy LaPalooza really adequately described, the land crab fishery here in the Bahamas is an important food source throughout the islands. And the tradition of harvesting crabs really carries with it a very vast cultural significance. Interestingly enough, though, with this significance, the black crab species itself is really understudied. Even the International Union for the Conservation of Nature listed as unevaluated. So there's some great voids in our understanding of this species. Next slide, there we are. So here on Eleuthera, we actually have 3 different species of land crabs. So there's the one that we mentioned already, the black crab, Gecoscinus rubricola. There's also the white land crab, Cartosoma guanhumi, which we were learning about. And also there is the red land crab, Gecoscinus lateralis. Of the 3 species, the black land crab is considered to be the most terrestrial, and this is given its choice in habitat. So whereas the white land crab is typically found in lower elevations, closer to the water table and is in wet burrows, the black land crab itself is actually forest dwelling and living within the coppice ecosystem. So to take a look at the coppice that we're talking about, this is a brilliant representation of the ecosystem that will find the black crabs. So what we're seeing here is an abundance of leaf litter, which is the crab's predominant food source. They are opportunistic omnivores. And you can see a very complex structure of limestone pitfalls and cracks and crevices. And this allows the crabs to be able to shelter themselves from predators and different environmental conditions throughout the day. The photo on the right really highlights the impact this coppice ecosystem has with its very dense canopy, which helps to block out sunlight as well as lock in moisture. And as we learned before, the crabs really need to maintain that moisture across their gills in order to be able to respire. Another strategy that the crabs implement is they're actually nocturnal. So other than during the running season when you may see them along the road margins, typically they're going to stay within that coppice and really only emerge after night. This was a sage piece of advice from the local harvesters. If you are going to do this kind of work, bring 2 flashlights. There is nothing worse than being out there and losing your light source. But yeah, and you can just kind of see the depth of this ecosystem, a lot of pitfalls, a lot of holes. So it's a really interesting one to study. This picture right here, I think really beautifully represents what we mean by these ecosystem functionalities that these crabs present. So here again, we see a picture of a limestone cliff face. And at the base, you can see it's pocketed with a variety of black crab holes. What we're seeing here is the brown soil is the excavation from these crabs. So they're actively aerating the soil and turning it over. And subsequently, by their feeding and bringing these leaf litter mass into the holes, they're actually working to sequester carbon. And in a 2009 study, a species of a similar species was actually described as being what's known as a biofilter in its respective ecosystem. So what does this mean, a biofilter? Essentially, in areas where the crabs are extremely prevalent and have high densities, they can actually act as a biomedium to essentially regulate which kind of plants will grow in that specific ecosystem. And again, the kind of continued story with this species is that we just don't have that information on the black land crab itself. So with the theme of this talk being food for the future and trying to understand what different threats these animals face, it's important to kind of go through these sequentially. So one of the topics we often hear in small island states is notions of habitat destruction. And this could take place for a variety of reasons, be it for real estate development or agriculture. And these are definitely not necessarily bad things. They're essential parts for food security and the economy here in the Bahamas. But it's important to know that with these animals, they really need these specific ecosystems. And if these are being destroyed, we really have to understand what that impact would be. Another threat with any kind of fishery important animal is the threat of overharvesting. Now, with the black crab and even land crab species in general, in order to understand how many crabs are being harvested to say whether or not that's sustainable, you really have to have an understanding of what that population looks like. And as Dr. LaPalooza had mentioned in her presentation, this can be an extremely challenging task. So this is one of the threats that we really do need to investigate further. Another one that we're really concerned with here is the presence of invasive species and what this might look like as far as predator-prey interactions, which we'll talk about in just a bit. And one of the larger scale themes in my research here is looking at climate change and how that's really going to affect the survivability of these crabs into the future. So as much as I would love to go through my entire master's thesis, I want to stay respectful of our time limits. So I've narrowed it down to about 4 of our main questions and goals with this work. The first is to really kind of assess the population structure of black crabs here on Eleuthera. The second is to look at how these crabs influence their environment or how they fit that role as an ecosystem engineer. Thirdly, we're looking at what predators do these black crabs face and looking at different characteristics of those predators. And finally, we're investigating how does climate change potentially affect these black crabs into the future. So as I had mentioned, I'm based in Eleuthera, Bahamas, currently down on the Southern Cape at the Cape Eleuthera Institute. And this ecosystem down here actually presents some really just great habitat profiles, but also the opportunity to see these different disturbed ecosystems as well. So the Cape has some really lush, deep coppice ecosystems, and it's punctuated by old disturbances such as airway strips and even golf courses towards the Cape. And you can actually see this fragmentation when you're on the ground. So in areas where the coppice is really dense and thick, you have an abundance of these black crabs, whereas in older or more recently cleared areas of the forest, yeah, you definitely don't see them to the same capacity. So with these goals in mind, the first idea is to start to assess the population structure and some various characteristics of these black crabs. So what we do is we've gone through at night to harvest the crabs. They are wild caught. And what we go through is do a variety of morphometric measurements. So we're looking at carapace width, carapace length, as well as the different dimensions of the biters. From this too, we're also very interested in the color morphologies that these crabs exhibit. So one of the first things that people talk about when they say black crabs is that though they have this namesake, a black crab, they can exhibit any kind of color structure from what we're seeing in the bottom left there, which is like this orange and blue white. I've seen them to be like these green kind of yellow colors. So it's really quite remarkable. And this is important because crabs across taxon really demonstrate color variety for a number of strategies, be it for mating cues or camouflage. And again, this is something that just really has not been explored with this species, both here on Eleuthera as well as throughout the Bahamas. With this, once the morphometrics are worked up and we've recorded that data, we're also doing a more traditional mark recapture survey. So the crabs will be fit with an individually labeled tag and released back into the coppice from their site of capture. With this, though, we're also interested in a little bit more of a fine-scale study about how these crabs utilize their habitat. So one of the ways we're doing this is actually through a partnership with Tile. If you're not familiar with Tile, they produce these Bluetooth signature tags, which traditionally are to help you find your keys, but now can help you find your crab. So these tiles, they work really well. They have about a range of about 150 feet in the copse ecosystem, at which point anybody with a Bluetooth-enabled device can read the signature from the crab, and this can really help us to isolate specific spots, be it burrows or caves underneath limestone or deep beneath the leaf litter layer. And especially now with current COVID restrictions, it even allows us to be able to do these surveys throughout the day as well, which is extremely important. So to highlight just a bit about how this works, here we have is one of the tiles that we first released. So you see there that little arrow marker is the release point. About a week later, we got a digital detection. So again, this is one of those Bluetooth signatures to the Tile app. Now, again, as I mentioned before, the range is about 150 feet. And what we're seeing here is that the radius, the GPS signal is taken right from the center of it. So from that center point, stretching out to 150 feet, and that's the range. When you combine this with repeat captures, so this was another Bluetooth detection signal, you can start to see a little bit of that crab motion. And now we're starting to be able to kind of hone in on where that crab occupies that space and that potential home range. And again, with a third recapture, another week later, you can start to see now that we have the capability to triangulate just a bit. One of the other really exciting things about this Tile technology is the fact that you can share these signatures with pretty much as many people as you want. And what this enables us to do is really engage with the local catcher community, as well as other schools and other programs. Again, anybody with a Bluetooth compatible device can actually live record this data, be it day or night, or just taking a run through the woods, what have you, which is really exciting. Another way that we're starting to look at how these crabs impact their ecosystem is we've actually gone through and have done an exclosure experiment. So within the study site, we have 10 replicate studies, 10 sites. They are meter-squared quadrats that are surrounded by a chicken wire fence. And to take a look on the image on the left, you can see the fence has a bit of a flared lip over the top. This is to ensure that the crabs can't actively climb into the pen. So what we do is on a bi-weekly basis, we go to all the study sites and we collect data on leaf litter depth, changes in percent coverage of leaf litter, as well as changes to the vegetation within the pens. So counting the new growth or seeing if things are being grazed upon. This study is being conducted throughout the year, and it's really exciting to see where this will go as we extend it into the future. Another portion of this study is looking at, again, the predator interactions that these crabs face. So pictured here, we have trail cameras that are posted throughout the coppice. In the bottom left, you can see a crested night heron, which actually has a crab in its beak. So we have an active predator-prey interaction there. But when we talk about invasive species and pressures in that regard, to take a look at the picture on the top left, you can see there is a feral cat. And this is one of our more common detections that we've been seeing. So it seems as if there is a quite numerous population of these cats down here in the south. And one of the things that's been the most surprising to us, it's been a huge talking point here on Eleuthera, is the invasive raccoons that have been making their way from north to south. So I'm sure other people have seen them in the coppice and around these ecosystems, but these might be the first photo evidence of the raccoons. And again, these camera traps are placed in active crab habitat, so it's likely to infer that there are some interactions. The next step is to actually put crabs of varying sizes into the field of view to actively see if there is that predator-prey interaction. So again, with all of this, a pilot study and plenty of work still to be done. And now to more of the main topic of the research that's happening here is implications of climate change, both in the Bahamas as well as throughout the Caribbean region, as it pertains to the prolonged survivability of the black land crab. So here's a 2013 study that uses a variety of climate models, all of which predict varying scenarios, but the common scenario and the common trend for the Bahamas tends to be one of increasing temperatures as well as decreasing amounts of precipitation. So looking at this 2013 study, to look up in the Bahamas region, we have a projected increase in temperature of 2.7 degrees Celsius by century's end. That's to 2080 to 2089, which, as any climate scientist will tell you, that is quite a significant jump, which is definitely concerning. In my opinion, one thing that's more concerning is the decrease in precipitation. So this figure from the same study is depicting how much of a decrease we'll see in annual precipitation. And again, to look to the central Bahamas region, projections are for a decrease up to 34% in annual precipitation. So this means overall more dry, less frequent rain events, and more consecutive dry days. And again, knowing what we do about crab physiology, knowing that they have to maintain that moisture across the gills, this has profound significance. So in the lab, we're actually simulating these drying conditions. Crabs caught earlier in the year have been housed at the CEI wet lab facility. They are fed in a brood stock, a variety of fresh lettuce, native leaf litter, and opportunistically some carrion as a protein source. And they have open access to water to maintain that hydration condition. In the situation where they're going to be going through the experimental dehydration, they're placed into the crab pen that we see on the right. And this was designed to maintain pretty much the same conditions that you would see in a crab burrow as it pertains to temperature and humidity. So the crabs will go through and go through their dehydration regimen in there. In an initial series of experiments, we really wanted to detect what was the maximum survivability of crabs in the face of dehydration. So the data that you see in this figure is presented as water loss as a percent of body mass. And we obviously have the box charts and the error bars as well. To take a look at this along the x-axis, we have the time intervals in days. And the experiment was carried out to the point where the crabs became what's known as moribund or unresponsive to touch, essentially incapacitated by dehydration. So from the mean values, one of the things that we detected was that crabs lose on average 1 to 2% of their total body mass daily, again, when not exposed to any moisture whatsoever. And after about 16 days, as I mentioned, this is where the experiment was terminated. This was about, on an average, a 18% reduction in body mass. And this is at the point where the crabs are, again, incapacitated by that level of dehydration. From this information, we then wanted to determine what does this effect actually have on crab behaviors, and specifically behaviors that are important towards these ecosystem service functions. So in the lab setting, we have a crab that's gone through its experimental dehydration. The one I'm about to play is a control. So it's 0 days dehydrated, and it has access to food and water in this experimental arena. One of the cool things about this is that the overhead camera, which takes a photo every 10 seconds over the course of 24 hours, actually has the ability to track the motions of the crab. So we're actually able to directly quantify the distance at which the crab has moved, the time that it's been spent either feeding or drinking, or sheltered in its burrow that you can see up above. Again, this is some preliminary data that's first coming in. We're still running these trials, but from some of the themes that we're seeing at this point is that the crabs with increasing dehydration levels tend to be exhibiting decreased activity levels. And again, knowing what we do about their ecosystem functionality, this has some profound ramifications for what these services are going to be looking like into the future. In regards to activity levels as well, what this means for the future of the fishery. Again, if these crabs are becoming increasingly more dehydrated, they may become increasingly more scarce for local harvesters. So that's kind of the 10,000-mile high explanation of the lab work that we have going on. The final component to this I'd really like to highlight is what does this fishery mean to local harvesters and to the local community? And I think the best way to do that is to talk with the stakeholders, or more specifically is actually to conduct surveys. So over the past couple months, we've actually been assembling stakeholder surveys with kind of a three-prong approach for the questions that we would be asking. The first is to understand how the fishery is utilized. Essentially, these are fisheries questions to identify potential catch rates of black crabs and to begin to identify the chain of custody and distribution of the crabs from the point where they're caught to the point where they're available on the market. And again, I think that this is going to be extremely varied from island to island based solely on the abundance of the black crabs, but also where people are in relation to the population centers. So again, as Dr. LaPalooza had mentioned, that typically in Andros, the crabs are shipped over to other family islands. The kind of the trend that we've been seeing here from personal correspondence is that both white and black crabs tend to be shipped further north up towards Harbor Island here on Eleuthera. So pretty interesting. Other questions will be geared towards trying to find out different market values and ultimately just trying to identify the economic significance of these crabs here on Eleuthera. The second component is looking at perceptions and attitudes of the crabs towards local Bahamians. So what does the fishery mean to local Bahamians? And what are some of the attitudes towards the fishery, whether or not people think that it is sustainable, whether the populations seem to be increasing or decreasing, and just to try to get a finger on the pulse of what local harvesters and stakeholders think about this fishery. And thirdly, we're really interested, again, in kind of tapping into this local knowledge base. When I first started this project, I knew very, very little about where to find these crabs, but you only have to ask a local harvester and they will point you to specific burrows. They will point you to specific roads where you can find the animals that you're looking for. So though we say there's a lack of scientific literature available, there is a plethora of local knowledge. And again, one of the goals of this survey is to actually start to channel that information to get it to one consecutive source. So, and with that too, the survey is also being modified too to represent COVID-19. Obviously the economic hardship that's going to follow and has insinuated from this pandemic, it's likely to infer that this fishery may be dependent upon further into the future, even in the immediate future. So developing questions around that as well. The next steps as well is also to get this project engaged with local citizens. So getting citizen science, working with catchers, and getting students involved with this project. It's extremely accessible, and it's something that, again, is representative of the local culture, both here in Eleuthera as well as throughout the Bahamas. And it would be a beautiful thing to get this project to be by Bahamians and for Bahamians. So it's one of our primary goals. Again, COVID-19 does throw a wrench in some of that social interaction. So definitely looking for suggestions on how we might be able to do this into the future. But I see I'm running over time, so I just have some closing thoughts. Again, this is a pilot study, and there is a plethora of questions that are still out there about this species, the black land crab. In order to make sure that we have this fishery and this resource into the future, it's my belief that it's paramount that we get this into the hands of stakeholders. So not just recording data to be sent back up to Newfoundland, but to retain it here in the community and have that community engagement. And based on the other discussions from this panel, I would be really interested to reach this out and get the other people involved. So if you in your line of work, or if you know of different programs are interested in collaborating with this project, be it through citizen science or taking a survey, I would encourage you, please reach out. I think it's paramount that we start to have a kind of a national network on understanding further aspects of this species. So with that, I have a few acknowledgements, primarily my supervisors, Dr. Ian J. McGaw and Dr. Nicholas Higgs. I would not be able to do this without your continued support and mentorship. Memorial University School for Graduate Studies for the funding of this project, as well as the Cape Eleuthera Institute and the Island School Outreach Departments for facilitating me here in South Eleuthera and for your partnership with this project. Again, a big shout out to Tile for their continued support and their generous donation of those tile tags. And last but certainly not least, Ms. Crystal Ambrose and her staff at Bahamas Plastic Movement. Your help in the field has been just paramount. I really appreciate that. So thank you very much.
All right, thank you so much, Mr. Bigelow. That was a fantastic presentation. And again, it's also a very concerning presentation because we're dealing with a very important fishery that is under threat due to climate change and over-harvesting. A second. All right, so we are about to get into the questions and answers period. But before we do, we hope that you can join us again for the remaining sessions that are scheduled to continue every Wednesday in August. As always, we welcome your questions, your feedback, and your support. As the BNT team continues to pivot our programming towards this new normal with new normal methodologies, including changing the platform of our conference to a virtual reality platform, there's still a need for funding events like these weekly sessions. We would be grateful if you consider donating towards the success of BNHC 2020. If you are enjoying the BNHC virtual webinar series, we simply ask that you consider giving a donation in any amount as a means of showing your support. An email with a donation link will be sent following this session. And so we are now moving into the question period. And I see that there is currently one question in the Q&A answer box. Just a reminder, everybody, if you do have any questions, you could just kindly just put them in the question and answer chat box. And this question is directed to Dr. Landry. Are there any organic or environmentally safe means to prevent attack on plants by the shield bug? And I guess the shield bug is synonymous with the stink bug. I just know those types of bugs to be stink bugs because they really smell.
I'm waiting because it says Giselle would like to answer the question live, and I'm not sure how to interpret that.
Oh, I think that she is also, it's because it's being streamed on Facebook. She was listening to the question live.
Oh, okay. Well, I guess the short answer is I don't know whether or not there is a way to do that. Shield bugs are stink bugs, and they do smell pretty bad if you irritate them. But technically, they don't smell bad until you irritate them. So the other thing I would point out is that I mean, if you have an insect infestation in your plants that are like your garden plants, food plants, etc., those are the times to be concerned about trying to remove herbivores. But if you're talking about, you know, your native garden or other areas, I would argue that one of the ways in which organisms interact with one another, like so herbivory on a plant of one species may actually reduce the likelihood that that particular species becomes much more dominant in the community. And so you could end up with greater biodiversity, or you do end up rather with greater biodiversity when you have things like herbivores and seed predators well, animal predators too, things that prey upon other animals, because it has a tendency to sort of knock down the population size of whichever component of that system is most dominant, right? So you increase biodiversity by having things that may damage plants sometimes, right?
Okay. Very, very true. Very interesting. Okay, so our second question is directed to Dr. Matthews, and the question is, have you been able to access the National Insect Collection to determine if there are existing records of plume moths for the Bahamas? Okay. Is Dr. Matthews available? No. Okay. Oh, she may be disconnected. So I guess we will move on to that question and come back to that question a little bit later. So this next question is, in the past, we would see hundreds of crabs heading across the roads towards the sea, but this does not seem to happen now. How will crabs continue to reproduce if they cannot reach the sea? I guess that can go to any of the crab experts.
So I guess maybe I'll take this one. And part of that is because when I was living on Andros in the mid-90s, we would actually still see, you know, these swarms of crabs literally crossing the road on North Andros to get to the sea. And I've definitely heard, you know, the elderly land crab harvesters talk about, you know, stretching nets across the roads to gather crabs. And so really it's, you know, there's definitely been a shift in the last 20 to 25 years. You know, when we're thinking about how, you know, how the crabs are going to be able to reproduce, you know, this reproduction is, you know, they'll be able to mate and to spawn, but it's incredibly important and imperative to their life cycle for the females to release the eggs into the ocean. There's about a 30-day part of the life cycle where these, you know, these eggs develop and are part of the pelagic network in the ocean. And so, you know, this is where having more knowledge about what's happening inland. So, you know, it's not necessarily the crabs can only spawn their eggs into, certainly thinking about Andros, that eastern edge, that, but really, what's happening inland. And so I do think that there's going to be reduced spawning in areas where the crabs are harvested pretty significantly. And again, the hope is that having this network of protected areas, that, the West Side National Park really becomes this space where, recruitment of crabs can occur because the crabs are spawning into those areas. And certainly like the local knowledge is important as well. The local harvesters will, if they've got a female crab in their crab pen that, is heavy with eggs, they will actually, they'll take the crab and they'll hold the female crab in the ocean to see if she will actually flip her eggs off of her abdomen into the sea and then go from there. So it's definitely going to be a challenge for reproduction, though certainly in these areas where there is heavier harvest during the spawning migrations.
Yeah, I also, it was amazing when I first went to Andros in 2007, and I saw, it was really amazing to see so many crabs crossing the street and just learning how to handle crabs and stuff like that. It was a treat. But yeah, we are noticing that there may be the population of adult land crabs may be decreasing as more and more people are harvesting, or more of the crabs are being harvested. And then also, I think what would be an interesting study would be to look at the how I guess the mortality of land crabs in those croaker sac bags as they're being shipped. If how much of those crabs are actually dying before they reach the city or those other places, because those animals are really packed in those bags, and it would be interesting to learn about the mortality of those crabs in the long term, and how that affects the population.
Yeah, and Scott, to really like to comment on the crocus sacs, and so there are people who have shifted. So instead of having the nylon crocus sacs, they're using more of the burlap ones. But still, really the challenge is the white crab has a much lower tolerance for dehydration than the black land crab, and so they're definitely, I think that there's definitely a higher loss when because people they'll bag their crabs really early in early in the morning to get them on the boat first thing. But still, there's still a period of 8 or so hours where desiccation can really impact the survivability just of that trip from the boat from, say, Fresh Creek to Nassau or to wherever it's going. So yeah, absolutely, Scott.
Yeah, that's always been a question for me, especially going on the fast ferries and seeing those so many bags of crabs, and I'm pretty sure that those crabs are not in the most comfortable situations in them bags. And then also it's hot, so they try to keep them in the shade, but even still, that's a lot of crabs being crammed up in that bag, and it is unfortunate. Okay.
Yeah, and I think that's where the distribution really comes into play too, is, to really improve the distribution to minimize that impact.
Yeah, most definitely. Okay, so this other question is to the crab experts. Have any guidelines been developed for crab harvesting, or standards considered as a means to protect the species?
Yeah, I can speak to what we've been seeing here on Eleuthera. And just from my understanding of the crab fishery throughout the Bahamas, it appears to me as if there's no government mandated regulations, but it does seem as if within the crabbing community, there is like a best catch practice and standards. So seeing a gravid female oftentimes will be left to, go through and go complete the spawning. But yeah, here in Eleuthera, it seems to be a little bit more artisanal, not to the same commercial capacity that you may see in Andros.
Okay.
And some of the earlier work, it's the land crabs are found, throughout the Caribbean down into, South America. And so there are some nations who have implemented some catch restrictions, and some of the earlier work that we did, looking at the sizes of the crabs that are being harvested, they're kind of at this mid-range. And so I think Brazil has a minimum catch size that's actually rather large. And Brazil also prohibits collection of females. And so whether that's happening in some of the local communities, I would gather that's still happening as well. But, no regulations, there's no catch size, but, there's definitely, I guess, what would be considered, for sure best practices. And so whether or not, the best practices are always followed, I think has shifted over the years as more people are traveling to Andros. Again, that's where I've spent the bulk of my time on this project, is really, the best practices exist, and I think are held true by the older generation of harvesters. But I think that there's become such an uptick in harvesting of this so incredible resource that those best practices might not always be followed by people that are just traveling to Andros, for a period of time. I've seen people sleeping in the bush for a week to simply just to collect crabs. There are a couple spots where, I was out collecting and I was like, is someone camping out here? And it was people living in the bush, living in their car, collecting crabs and then sending everything back over.
Wow.
So it's definitely the crab harvesting culture and the fishery itself on Andros, I think, is, unique to the few islands, because I've been on about a half a dozen islands with this project. And Andros, Andros is the big yard and there's a lot of habitat for land crabs. And so I think that shifts what is happening with regards to harvesting this resource.
That is very, very true. Another question is, this one's directed to Dr. Landry, and that is, what species of insects were most commonly seen pollinating Turnera and Erithalis? And is there any insect that uses those two species as a host plant for their larvae?
Sorry, I forgot to unmute. The primary pollinators I'm still working out, because remember, it's not just whether or not the insects visit the flowers, but whether or not they effectively transfer the pollen to the appropriate location in the flower. I mentioned that Xylocopa cubicola, which is the big black carpenter bee, is very efficient at pollinating Turnera. I was able to follow those bees to a number of different flowers. I don't have that data right here with me to tell you exactly how many, but flowers that were visited by Xylocopa, the portion of the flower that gets the pollen deposited is called the stigma. And Xylocopa was as good as I was at covering that stigma with pollen grains. So, I was pretty good at it because I got 80+% fruit set. They're pretty good at it. In terms of Erithalis, that has been a little more challenging. The Erithalis diffusa sits no more than about a foot off the ground, the whole plant. And so tracking the movement of pollinators, you can't really check and see if there's pollen on the stigma. You have to go with, does it set fruit after the visit is made, as a way to determine whether or not, which individual species would be effective. And so I haven't got particularly any, like a main pollinator. I would say main visitors include the two bombyliid flies, Chrysanthrax, and what's the other one that I think Anthrax insular, which I don't believe was on my list because that was from a different study. But they have a regular, they are very common visitors to those flowers. And if you've ever watched the bombyliid flies, they're the ones that kind of look like bees almost. They're the ones that look kind of like cigars. They tend to stay near the ground when they forage, because that's the thing is with different pollinators, even insects, you think they might be anywhere, but really there is a a difference in where some species tend to be found in terms of their foraging behaviors. And the flies tend to stick lower to the ground. In terms of host plants, I have read but have not yet seen the Mexican fritillary in other parts of the Turnera range. The Mexican fritillary uses Turnera species exclusively. Debbie may be able to, Dr. Matthews may be able to speak more to that. She knows a lot more about butterflies and their larval food requirements than I do. But that's what I've seen in the literature. I have not actually found Mexican frit larvae on the plants, nor seen butterflies doing laying behaviors on Turnera. Yeah, so that's the short answer. And Dr. Matthews said she's back.
Yeah, I'm cut out twice, but I'm here. I'm not really familiar with the life history of the Mexican fritillary, but that's something we can certainly look into.
Okay. Okay. So the other question is to Dr. Matthews, and that is, are the larvae of the plume moths toxic?
Not that we know of, but you may have noticed that there are some species with the glandular setae, and it's a very sticky substance. We don't know what it is. One thing I'd like to do in the future is maybe some bioassays with ants, just to see how they interact. But it's very, the exudate is very sticky, frass and pollen, everything sticks to it. Some people have hypothesized that, it could be something to deter predators, either chemically or just, tanglefoot. So, yeah.
Okay. Okay. Interesting. Mr. Bigelow, this question is for you. Do you know if the black land crabs change their color as they get older?
This is a great question, and one we're actually very interested in exploring. So from what we see, the color varieties are all over the place. The vast majority seem to have, a darker kind of complexion. One of the things we've seen in the lab is that there actually does appear to be a bit of plasticity in the color variation, specifically with the lighter colored crabs. So long story short, I don't believe that there's been any studies that have looked into that specifically, but it's something that we're very much interested in exploring further.
Okay, and a follow-up question would be, I think you may have touched on it in your presentation, but do you have an idea of what the home range of a black land crab is possibly?
Yeah, so given the life cycle, obviously the crabs are subject to their seasonal migrations. But again, this is just another one of those areas where with this species, it really hasn't been explored to great depth. Now, you can infer from other pieces of literature about similar carcinid species, and there is evidence to suggest that they do kind of occupy a home range. Now, whether or not that's a specific burrow similar to that, maybe of like the white crab, is hard to say. But yeah, it's something we're actively investigating currently.
Okay, thank you so much. And this is another crab question for Tammy, and that is, do you have any other plans to conduct land crab surveys on Andros, namely South Andros?
Yeah, so it's been a while since I've been to I was on South Andros for the initial study, I think back in 2009, when we were working on the genetic component of the project. I think South Andros, I don't have in the pipeline a grant to get funding to get to South Andros. However, that's definitely on my list. One of the challenges is certainly right now travel is pretty much banned, as is approval to go to conference or to leave to go conduct research. But yeah, I was on South Andros for, I think, under 72 hours back in 2009. And that's definitely a spot where a survey of the populations there, I think, would be incredibly important. Just from some of the conversations I've had, the populations there are possibly even more robust than that on North Andros. And probably also protected a little bit because there's not, there's seasonal influx of tourists that travel to North Andros for Crab Fest, which certainly, impacts the populations as well. And so that large influx, 20,000 people in a weekend, and how many of those are actually active harvesting crabs, I'm not sure. I know people go to Crab Fest only to harvest crabs. But yeah, South Andros is definitely on the top of the list for another crucial site to really start looking at the population structure and what's happening within those habitats.
Okay.
Yeah, if anyone's interested in to collaborate to get to South Andros, let me know.
Will do. We'll let you know.
I'd love to get back there.
I hear you. So we got about 5 more minutes. This is another question. The question is, has anyone encountered centipedes that also thrive in moist places to be a threat to crabs, particularly when spawning? I guess let's go to any of, go to either Tammy or Mr. Bigelow.
Yeah, so when I was first researching, the background literature review of the black crab, one of the first papers and one of the few papers is actually looking at different interactions with various flies that also live in the burrow. Now, unfortunately, I can't really speak specifically to centipedes, but one of the things with specifically the black crabs is there's this notion of these lost years. So when these juvenile or the megalopa return from the ocean, it's anywhere from about like that first to like the 4th year of their existence where you really don't see the juvenile black crabs. And the running theory that we're trying to prove at this point is to see whether or not they're occupying the burrows of adults. And I could imagine if there were centipedes in there, that would be an active predation on that. That's a really compelling question.
Okay. Very, very interesting.
Yeah, and I've read the same with the white crab as well, flies. And there is literature out there that does indicate, even for the white crab, that there can occasionally be a smaller juvenile crab that is basically taking up residence in an adult land crab burrow.
Very, very interesting. Okay. So I don't see any other questions.
There was a question for.
Oh, yes,
Dr.
for Dr. Matthews. Right.
Oh, okay.
And this final question is, have you been able to access the National Insect Collection to determine if there are existing records of plume moths for the Bahamas?
Yeah, we not with plume moths. I had the opportunity to look at the collection in the College of the Bahamas, and Jackie has investigated the national collection, and there are no specimens there. And even if you were, you have to understand that these are so small that it the slightest bit of damage could be a problem. So I can't emphasize enough the need for tight fitting drawers and pest control. Yeah.
All right. Yeah, we definitely need to do a lot more work in that department. Okay, so I just want to thank all of the researchers for presenting today. This was another fantastic webinar, and we are so grateful to have you guys. And again, we are having another webinar on Wednesday next week, and so we hope that you all can attend, and we look forward to seeing you all there. If there's any more questions? Let's see. Okay, so there's.
I would say that the National Collection has good cabinets with tight fitting drawers for the insects.
Well, that's good to know. Awesome.
There's more to it.
For sure. I've only
There's more to it than that, but at a minimum, that is true.
Okay. All right. So once again, thank you all so much, and we do hope to see and hear from you guys in another webinar presentation that the Bahamas National Trust will be leading, hopefully in the not too distant future. So in all, thank you all, and take care.
Thank you, Scott.
Thanks, Scott. Thanks, Giselle.
Thanks, Scott and Giselle. Have a good afternoon.
Yep. Everybody have a good day.
Yep.
Thank you.
Bye-bye.